So the work that we do, I guess we can say it focuses on the one and the many. The one is E. coli, the bacterium that we're most interested in, and many people have heard of E. coli. It is actually very commonly used in scientific research as a model organism, but it's also a very versatile pathogen and can cause a wide range of diseases in humans and in animals. And what we're interested in, though, is a rather neglected field, which is how does E. coli live in its natural habitats, not in the laboratory, but when it's living in people's guts or in the guts of animals. How is it actually making a living? And what are the rules that allow it to live there? How do different strains of E. coli either live together or compete with each other? And our motivation is we're curious about these things, but there's also an impact, which is that what we want to do is repurpose E. coli and turn it into what we might call a probiotic, a kind of friendly bacterium that can be used to compete perhaps with pathogens, the dangerous varieties of E. coli, but also deliver additional benefits on top of that. And so understanding the rules by which, say, different strains of E. coli compete with each other, how they can live together, and so forth is one of the goals of that research there. We're also interested in E. coli's role in disease, and we're looking at how E. coli can become resistant to antibiotics, and we have a study with a collaborator in Pakistan where we're looking at multi-drug resistant varieties of E. coli and trying to work out how they've become resistant over time. So in studying E. coli, we use a range of techniques in the laboratory. We grow the organism, we do genetic manipulation. But a key feature of the research that I've been carrying out over many years is actually sequencing genomes, so actually determining the DNA content of the cell and working out the letters in the alphabet of DNA and the sequence of those within the organism. And that can give us great insights, not just into how the bacterium can cause disease, how it lives, but also how it's spreading and how it's evolving over time. So that's the one. The many is that we also look at microbial communities. So humans and animals are home to very complex microbial communities. We sometimes call these microbiomes. We have this very complex microbial community in our own gut, particularly in our large intestines. And this can be very important in determining the balance between health and disease in humans and in animals as well. So one of our focuses is looking at the gut microbiome in critically ill patients. So these are patients in intensive care unit who've had many different antibiotics and many other physiological assaults. You know, they've been very ill, they're given acid suppressant medication and all sorts of other things. And what we want to look at is what happens to their gut microbiome in that circumstance, and understanding how bad the damage is, and laying the groundwork so how we could then manipulate that to help restore health or maintain health in those critically ill patients. We're also doing similar work with celiac patients when they get diagnosed and then put on a gluten-free diet. We're looking at what happens to their gut microbiota, the gut microbiome, and how that is helpful in restoring health or where it might be an impediment to health. In addition to that, though, the main theme over coming years is going to be looking at food animals and looking at the microbiomes of food animals, in particular the chicken. And like humans, the chicken is home to a very complex microbial community in its gut. But we understand very little about that. There are, I think it's 9 billion chickens across the planet. But we understand more probably about the dark side of the moon than we do about what lives inside the chicken gut. We've done some studies already, and we've found that probably half the bacteria that we find in the chicken gut are unknown to science. They haven't been given names, they've not been seen before. And so actually just characterising what lives in the chicken gut and getting a list of all those organisms, characterising them, sequencing their genomes, is going to be a major task for the coming years. And we'll also lay the groundwork for looking about how that microbiome, those genes, genomes, germs, vary under different conditions, because this is crucial to the health of the chicken, the balance between health and disease in the chicken, their welfare, but also to productivity in the food chain as well. So that's going to be a key theme.